Hey guys, this is Malinki. Welcome back to my channel, Voice of Malinki. Today we will talk about cell cycle and cell division, especially mitosis. And if you are new in my channel, please subscribe my channel. And if you like my video, please do like, comment and share my video. So let's talk about cell cycle and mitosis. Okay. So this is the cell that is laughing at you. So this cell is dividing. This cell is dividing into two cells. So this is the parent cell or mother cell. And these two are daughter cell. Now, these two daughter cells again undergo division and they divide into these four cells. At that time, these two cells are parent cell and these four cells are daughter cells. Like these, cell division occurs and from one cell, million of such cells are produced. So, any organism starts from a single cell like this. This is the single cell. And you know what is that cell? That is the zygote. That is the zygote. This is the single cell and from that single cell, any organism develops. So when this single cell divides and generates millions of cells, this is the organism that is generated. So, we need to understand cell cycle first. Now, cell cycle is a series of events that takes place in a cell as it divides. So, a cell divides and generates number of cells and many events occur during that time. That is called cell cycle. Now, a cell cycle has different phases. First of all, in a broad scale, the cell cycle is divided into two main phases. First phase is the interphase and second phase is the mitosis phase. These are two phases, main phases which occur in cell cycle. Now, interphase is further divided into three stages: G1 stage, S stage and G2 stage. G1 and G2 are called gap phase or growth phase. And the S phase is called synthesis phase. So these three phases are altogether called interphase. And the mitosis phase, or you can say it M phase, this phase is divided into two main phases: karyokinesis and cytokinesis. So, now a human cell takes 24 hours to divide. Suppose this is the human cell and this is the parent cell. This cell divides into two cells. These two are daughter cells. Now, it takes 24 hours to generate these two cells. So, cell division takes 24 hours and this is for human cell because for every cell it is different. Like in case of yeast, it is only 90 minutes. So yeast cell takes only 90 minutes to generate 2 cells, or to complete its division, yeast cell takes only 90 minutes. Now let's talk about human cell. Now, among this 24 hours, interphase itself takes 23 hours or 95% time. 95% time or 23 hours is taken by interphase. And a mitosis phase only takes 1 hour or 5% time. Okay. Now this interphase that takes 95% time, that is actually preparatory phase. That means it is the preparation. It is the preparation. During that time, cell makes all the necessary components which it requires for its division. That's why this is the preparation phase. When you cook something, it takes more hours for the preparation. But the cooking is very fast if everything is there in your hand. Like that. This is the preparatory phase that takes 95% time. And the mitosis is actual cell division phase. When the one cell divides into 2 cells, that is called mitosis phase. Right? Okay. So we have seen that interphase contains 3 phases. First one is the G1 phase. That is the gap phase or growth phase. Now, in this picture, you can see that G1 phase is between S phase and M phase. This is M phase. So G1 phase is between M phase and S phase. Now, G1 phase, during this phase, cell grows. Suppose this is the cell. This cell decides to go through the cell cycle and it will divide. So this cell, what it does? It first grows like this. So the volume of the cell increases. The cell grows, and that is the thing that occurs in the G1 phase. So in G1 phase, cell grows. The size of the cell increases. Next phase that we have seen in this picture, that is S phase or synthesis phase. So after G1 phase, S phase occurs. Okay. Now, from the name itself, that is the synthesis phase, you can understand that DNA replication or DNA synthesis occurs at that time. That is the S phase. Because when a cell divides into 2, the new cells will get new DNA. That means the DNA should be replicated before cell division. Otherwise, new cells will not be able to get the DNA. So, DNA replication or DNA synthesis occurs during S phase. Okay. Now, see what happens. So, this is the condition of a cell in G1 phase. So, you can see this is the cytoplasm. Here, this is the nucleus. And in the nucleus, you can see chromosomes. So, we human have 46 chromosomes, and I am representing here only 2 chromosomes. So here we have 2 chromosomes, right? And this is the centrosome that is present in the cytoplasm. So this is the condition of G1 phase. Now, this cell is now entering S phase. And I have told that in S phase, DNA duplication or DNA replication takes place. So you can see that one chromosome here contains one double-stranded DNA, right? Now here, this double-stranded DNA is replicated, and you can see here that 2 double-stranded DNAs are formed here. Now, don't call them chromosomes. These 2 are not chromosomes. These 2 are called sister chromatids. So these 2 newly generated DNA are now attached to this centromere, and they are not at all detached. They are attached, so they are not chromosomes. They are called sister chromatids. Now, 2 things you need to remember here that is very important for your understanding. Suppose I am talking about a human cell where we have 46 chromosomes and we have diploid set of chromosomes. So we have 2n. 2n number of chromosomes we have, 46 chromosomes we have. That means n is equal to 23. So we have 2 sets of n or 2 sets of similar chromosomes. So we are diploid. We have diploid cells. Now, in this case, after S phase also, cells remain in this 2n condition, because here also only 46 chromosomes are there, but their DNA duplicated, but those DNA, 2 DNAs or 2 chromatids are now attached with the centromere. So cells remain in the 2n condition, but one thing is changed. That is the amount of DNA. Suppose the total amount of DNA in one set of chromosome is C. And that is one set of chromosome means in 23 chromosomes, we have the total amount of DNA that is suppose C. So we have diploid set. So 2n is equal to 2C. N is equal to C. So 2n is equal to 2C. Now the DNA duplication occurred. That means now we have double amount of DNA. So at that time, we have the DNA amount that is 4C. So we have the amount of DNA that is now 4C. The amount of DNA is doubled in a cell. But the cell is in the diploid or 2n condition. Is this clear? This is very important to understand because many questions come from these 2n and 2c, 4c parts. Next phase is G2 phase. So we have seen here that after S phase, G2 phase comes. Now in G2 phase, mainly protein synthesis occurs as well as cell grows in size, and mainly here protein synthesis occurs because you have seen here that after G2 phase, the mitosis phase or actual cell division phase will occur. So, cell division itself requires many proteins, and those proteins will all be synthesized in the G2 phase. And in this phase, the cell again grows in the size. Now, the main cell division or mitosis phase will occur. That is the M phase. And we have seen that M phase is divided into 2 phases. One is the karyokinesis and second is the cytokinesis. So karyokinesis is nothing but the division of nucleus. That means one nucleus, suppose this is the cell and this is the nucleus. So one nucleus divides into two. So this is the nuclear division, that is karyokinesis. And this nuclear division or karyokinesis, it consists of some steps. First one is the prophase. So here we can see what occurs in prophase. This is the first step of mitosis. Here condensation of chromosomes occur. Actually, before prophase, in the interphase, what happens? Chromosomes were in the thread-like structures, like these, loose thread-like structures. Now condensation of chromosomes occur and the chromosomes become condensed and they are like this. So the condensation of chromosomes occur and from the thread-like structures they become condensed. Now chromosomes become untangled. So DNA replication has taken place and DNA is well like in this form. And now they are untangled. So the DNA becomes this. Third, centrosomes move toward opposite poles. Here, centrosome you can see here, this is the centrosome, and here this centrosome has already divided. So I think I forgot to mention here that in the S phase, it was the centrosome that was present in the cytosol and this centrosome is divided into two. So in G1 phase we had only one centrosome and in S phase it is duplicated, it is divided into two centrosomes. And please remember one thing, centrosome is only found in the animal cell. Centrosome is not at all present in the plant cell. Okay, so two centrosomes are now produced in the S phase and in this prophase, these two centrosomes, they move toward the opposite poles of the cell. One centrosome is here, another centrosome is here. They are now moving toward the opposite poles. Aster formation occurs. What is aster formation? So, from this centrosome, you can see, if this is the centrosome, you can see like these structures are coming out from this centrosome. These are called asters. They are actually some microtubules which are radiating from the centrosome. That is called aster. So aster formation occurs. Spindle fibers appear. So these are some spindle fibers. These are spindle fibers. These are also structures which are made up of microtubules. And aster and spindle fibers altogether we call them mitotic apparatus. So this aster and spindle fibers we call them mitotic apparatus. Golgi complex, endoplasmic reticulum, nucleolus, nuclear membrane disappear. All of these are now disappearing. So you have seen that in the S phase, you have seen this is the nuclear membrane. Now here you can see the nuclear membrane, what was supposed to be here, is now vanished. It does not have any nuclear membrane. And endoplasmic reticulum, Golgi complex, nucleolus disappear. Next is metaphase. So in this stage, nuclear membrane is gone. It is gone. Now what will happen? The chromosomes are now spreading through the cytoplasm like this, because nuclear membrane is not there. So chromosomes are free. They are free to float around the cytoplasm. So they are now spreading through the cytoplasm. chromosomes are most condensed. So now at this stage, chromosomes are very much condensed like these. They are very much condensed, and that time they are clearly observed under microscope. So if you want to see the morphology of chromosome under microscope, metaphase is the best phase where you can observe it clearly. Sometimes in our practical laboratory, we need to check the chromosome structure. That time, definitely, we will see the metaphase stage so that we can see them properly, because as they are most condensed, these chromosomes take stain very properly and we can clearly see them under microscope. Spindle fibers attached to kinetochore. You can see chromosomes are now at the center of the cell. That is called metaphase plate. That center contains a imaginary plate that is called metaphase plate. Here chromosomes lie at the equator. All chromosomes are now lying at the equator. And you can see that spindle fibers which are coming from the centrosome, these spindle fibers, they attach to the kinetochore. So what is kinetochore? Kinetochore is actually a structure that is found in the centromere. In the centromere, it has a particular region where some proteins are there. That part is called kinetochore, and in the kinetochore, the spindle fibers attach. So kinetochore is basically in the centromere. So now spindle fibers attach to the centromere. Now next phase is anaphase. So each chromosome is now split. You have seen in the last picture in the metaphase that these are the sister chromatids, right? And now in the anaphase stage, each chromosome is split and the sister chromatids are getting separated. And spindle fibers shorten. You can see here these are the spindle fibers which are now short. So they are shortened and pull two sister chromatids. These short spindle fibers, they pull two sister chromatids towards two opposite poles, because during metaphase stage, sister chromatids were like that. Now the sister chromatids are getting split and they are pulled by the spindle fibers to the opposite poles of the cell. That time, this is very important. At that time, the chromosome are not in the diploid, that is 2n, but they are in the 4n form, because these are not sister chromatids now. These are individual chromosome. Because they are not at all attached to each other, individual chromosome is forming. So from 46, you will get 92 chromosomes now. When it is 92, so the cell is in the 4n condition. This is very important point. You have to remember that in anaphase stage, the cell is in the 4n condition. Next phase is telophase. In telophase, decondensation of chromosomes occur. So now again, the opposite things are happening. After condensation, decondensation is occurring. Cells, chromosomes are again becoming thread-like structures. Nuclear membrane develops. You can see the nuclear membrane is developing. Nucleolus, which was missing, endoplasmic reticulum, Golgi complex, all are now again formed. So these are the things which are happening in telophase. Next is cytokinesis. So prophase, metaphase, anaphase, telophase. These four structures are found in the karyokinesis, and next phase is cytokinesis, because in telophase you can see nuclear division occurs. Two nucleus are now generated, but these two nucleus are now in the same cytoplasm of the cell. Now this cytoplasm also should be divided to generate two new cells. That will occur in cytokinesis, that is the division of cytoplasm. And the division of cytoplasm or cytokinesis is little bit different in plant cell and in animal cell. So here this is the animal cell. Now what happens here? Here a cleavage furrow occurs in between these two nucleus. It is the cleavage furrow, and this cleavage furrow will come deeper in this direction, and then it will split the thing and two cells occur like that. At that point the chromosome was in the foreign condition. Now cytokinesis occurs and each cell contains 2n chromosome. This is very simple thing that happens in animal cell. And now in plant cell, actually in plant cell, the problem is the plant cell contains cell wall. And the cell wall was not present in animal cell. Animal cells are delicate than plant cell. So the cleavage furrow is not occurring in the plant cell like the animal cell. In this case, the cell plate is rather formed here. So cell plate, what it does, it actually grows at that direction, and this is the cell wall you can see, and the cell plate moves toward the cell wall and it meets the cell wall, and that time it divides the system into two cells, and this cell plate, it becomes middle lamella. After the division of two cytoplasm, it becomes middle lamella that is present between two plant cells. So this is the primary cell wall. This is the primary cell wall of one cell, and this is the primary cell wall of second cell, and this is the middle lamella that is present between two cell walls of two cells. So at that time again the chromosome becomes 2n if it is the diploid set. Last thing that is also important, that is G0 phase. G0 phase, or this G0 phase is also called as inactive phase or quiescent phase. Quiescent, quiescent phase. I think the spelling is correct. So this is the G0 phase. Now what happens here? Some cells, you know, which are continuously dividing, which are very active cells, they will go through these stages which I have taught. But some cells are there which do not divide at all, like nerve cells or neurons and muscle cells. These cells do not divide. What they do, these cells, after their production, they go to the G0 phase. At that phase, these cells remain metabolically active. They are metabolically active. That means everything is occurring in the cell because the cells are alive, but they do not undergo any cell division. So they are in the G0 phase or quiescent phase. Now some cells are there who always do not divide, but if required, they will divide. Suppose kidney cells, liver cells, these cells will not always divide. But if accident takes place, if some cells are lost, if the infection occurs, if the organ is injured, that time those cells are undergoing apoptosis and new cells should be formed. At that time, what they do, the cells which were in the G0 phase, which were in the inactive phase, they will come into the G1 phase again and they will start cell cycle. And after some rounds of cell cycle, when those cells replenish the old cells or the lost cells, what they do, they will not undergo any such division. So again they will exit the G1 phase and they will enter the G0 phase and they will remain in the quiescent phase. Again, if required, they will enter G1 phase and they will continue the cycle. That is all about cell cycle and mitosis.